Wednesday, everyone. Thank you all so much for tuning into my first ever Women in STEM Wednesday. Please hit the subscribe button below so that you can stay up to date on all of the fun, exciting activities and content I have to release in the upcoming weeks. I'm really excited about today's video because I'm going to be sharing with you all information about one of my favorite female scientists ever, Rosalind Franklin. Rosalind Franklin was the first person to ever discover the structure of DNA, which is a double helix. And she did this through a method called X-ray crystallography. Unfortunately, two other male scientists named Watson and Crick found her notebook and took her discovery without giving any of her credit and then published it. And they won the Nobel Prize for this work. So she's often thought of the woman who should have gotten the Nobel Prize but didn't. But I'm so happy that to this day, people are still giving her recognition for her awesome discovery and for being an impeccable woman in science. So I'll start off today's video by first telling you why I think DNA is so important and fun to study. I'll then read you all a story about Rosalind Franklin's life and her research, and then I'll finish by teaching you all a fun and tasty activity that you can do at home that helps you remember what the DNA structure is like. So DNA is often thought of as the building block or the blueprint of life. Nearly every living organism has DNA, whether it's a human, an animal, or a plant. DNA is what makes all of us unique. Approximately 50% of our DNA is inherited from our mother, and then roughly the other 50% is inherited from our father. And because of this, we can actually all think of ourselves as mutants, because we're neither a direct copy of our mother nor father's DNA. DNA is used in so many techniques and applications that let our world go round. As a plant geneticist, I see firsthand the importance of DNA because I study the genetics of plants to try to improve them, try to make them higher yielding, more resistant to disease, so that we can ultimately provide more food. But that's not the only reason why studying DNA is important. DNA is extremely important to the medical industry. In fact, one of the first GMOs, or genetically modified organisms, was a bacteria that produced a lot of insulin. And they did this by modifying the bacteria's DNA. DNA has been used to develop numerous vaccines. It's been used in nutritional research. It's been used in cancer research. Right now, DNA is what's needed to test for coronavirus. DNA is also used in forensics. One of my favorite shows is Forensics Files. Over time, DNA has been used to convict criminals who have committed horrible crimes, while also proving some people innocent who might have otherwise gone to jail or faced a lot of punishment for a crime they did not commit. Right now, something that's become really popular is Ancestry.com, 23andMe, things that basically look at our compilation of DNA using something called markers, and they try to help us determine what our ancestry might look like. These are just a few of the many examples of how DNA has played a role in our modern society. Today I'll be sharing one of my favorite books of all time with you all. It's called Women in Science, 50 Fearless Pioneers Who Changed the World, and it's both written and illustrated by Rachel Ignatowski. If you haven't heard of this book, I definitely recommend purchasing it for yourself, if not for a friend. It makes an excellent gift. I actually got it for Christmas. And what you'll see is it is beautifully illustrated. It talks about the stories, lives, and discoveries of 50 amazing women in science from all fields, whether it's physics, chemistry, math, genetics, biology, zoology. And today I'm going to read you the excerpt and story about Rosalind Franklin, so you'll have a little bit of a background on why she was so important. So Rosalind Franklin was both a physical chemist and an X-ray crystallographer, and that was the technique she used to discover the DNA double helix. So I'll quickly read her excerpt before we get carried away with our fun activity in just a little bit. Rosalind Franklin was born in 1920 in London. Her father wanted her to do the work that he considered fit for a lady. He disapproved of women going to university. The women in Rosalind's family helped her stand up to her father. She went on to earn her PhD in physical chemistry from Cambridge University. The big question of the day was, what is the shape of DNA? Scientists knew that DNA formed the building blocks for the body, but they had no proof of what it really looked like. Rosalind Franklin was one of the scientists at King's College who were on the case. Rosalind spent hours and hours using an X-ray on the delicate fibers of DNA. She captured the famous photo that proved DNA is a double helix. Meanwhile, two scientists, James Watson and Francis Crick, were also trying to figure out the structure of DNA. They snuck a peek at Rosalind's work without her permission and used her findings to publish their own work without giving her any credit. As a result, she was overlooked. Rosalind left the toxic work environment of King's College and continued her research. She went on to a top research lab and started doing interesting research with the tobacco mosaic virus and the polio virus. Unfortunately, Rosalind was diagnosed with terminal cancer, probably caused by the radiation from her dedicated work with the X-rays. She died in 1958 at only 37 years old. James Watson and Francis Crick won a Nobel Prize after Rosalind died. James Watson wrote scathing, tasteless comments about Rosalind in his book, The Double Helix. He also admitted that he had looked at her data, and the people started to figure out how the discovery really happened. Rosalind is remembered as a woman who should have won a Nobel Prize. Now that we know the story of her groundbreaking work, we can celebrate all that she has accomplished. And that is exactly what we are about to do when we create our own DNA candy models. Today we'll need 3 things to make our candy DNA model. The first are Twizzlers. Any color will do. I decided to get some fun sour and sweet Twizzlers for today. You'll also need toothpicks, and then you'll need something to represent the bases, which I'll talk about in just a little bit. These can be dots or gumdrops or gummy bears. Before we start making the model, let's talk a little bit about the structural components and what these 3 things represent. The first thing you should know about the DNA structure is everything is held together by a backbone, and this backbone is called a sugar phosphate backbone. We're going to use Twizzlers to represent it. Also, perhaps the most important part about DNA are the bases. There are 4 nucleotide bases in DNA: A's, T's, C's, G's. Since DNA is double-stranded, it's important to know what bases pair together. A's pair with T's and C's pair with G's. We'll keep this in mind when we're picking the colors of gummy bears, dots, or gumdrops to use. Finally, we're going to need something to hold these 2 complementary bases together. Today we're going to use toothpicks. Now let's get to assembling. As I mentioned previously, it's really important that our bases are complementary to each other and that we're pairing A's with T's and C's with G's. For today's purpose, green gummy bears are going to represent A's, yellows will represent T's. Reds and whites will also represent C's and G's, respectively. So the first thing I'm going to do is use my hydrogen bonds to bind together an A with a T. And again, you can use whatever colors you want for your different nucleotides. Just make sure that you have 4 different nucleotides for the A's, T's, C's, and G's. I've already combined G's and C's and some other A's and T's together just to save time. So we have our sugar phosphate backbone, and we're going to take our nucleotides and stick it between our phosphate backbone because our phosphate backbone is what holds the entire structure together. And I'm just doing alternating A's, T's, and C's and G's. This is going to be so yummy. I cannot wait to eat this. I am the hugest fan of Twizzlers and gummy bears. So what we have right now in our hands is just 2 strands of DNA bound together that are complementary. But if we want, we can make it look more like the double helix structure that Rosalind Franklin discovered by simply twisting it into a helix shape. I hope you all had a great time today learning about an impeccable female scientist. Stay tuned for next week's episode where I'll be discussing yet another amazing female scientist.